Well, I find this a very interesting gathering together. I mean, there are many topics, but this is a nice thing about this, because usually when we go to meetings and we see the same people, we know exactly what they are going to tell us, and it's kind of boring. And today, every lecture is different, and you can learn new things. Okay, so the scope of my presentation will be, I'll tell you about the TRP channel, the origin of the name TRP that I call it. And you'll see it looks like a very narrow topic, but actually it's not. It's a very wide topic. And then I'll tell you a little bit, because most of our work was done in Drosophila photoreceptors. And I think it's not accidental, because the nice thing about Drosophila is that in the research you use unbiased approach. You screen for mutants, you have no idea, it's a random process, and look at your phenotypic expression, and you are bound to find new proteins and new processes. And this is very true, because this is a completely unbiased approach. And then I'll talk about the TRP channel superfamily, which is a polymodal channels which are activated either directly or via transduction cascades, and they are involved in almost every aspect of our life. Yeah, I'm not exaggerating. Then I'll tell you more about recent experiments about structural features of the TRP channel, because very recently the structure of the TRP channels was solved in below 3 angstrom resolution. And this is quite revolutionary, and this helps a lot to understand these remarkable channels. And finally, because I saw that in the program there are many talks about neurodegenerative disease, so I'll show you that TRP channels, or the American call them TRP channels, they are also involved in neurodegenerative disease. And maybe they can explain brain damage, ischemia, and things like this. So I'll talk about activation of TRP channel with anoxia, which is relevant. So how it is all started? So it is all started when I came to the lab of Bill Pack, and at that time, molecular genetics and molecular biology were in a very early stage. And most of the work was to isolate mutant. So every PhD or postdoc has to choose a mutant to work on. And to the disappointment of Bill Pack, I chose to work on a mutant that was spontaneous mutation, and he didn't produce it, so he was quite disappointed. But I was intrigued by the behavior of this mutant. It was found in Scotland, and in dim light it behaves normally, but if you increase the light intensity, it becomes behaviorally blind. So I was intrigued by this, and this was reversible. So I was intrigued by this phenotype, and did, with the help of a PhD student, Chung-Fong Wu, who is now in Iowa, and he was expert on intracellular recording, so I learned from him how to do intracellular recording. And here you can see an historical recording, intracellular recordings in a single photoreceptors of wild type and this mutant. They call it the A mutant in Scotland. Probably they thought there would be a B, C, and D, but there was only A. So if you give a continuous light in wild type, there is a sustained depolarization. You know, invertebrates, they depolarize, unlike our eyes that we are hyperpolarized. In the mutant, if you give dim light, it still has similarity to wild type, but if you increase light intensity, the receptor potential declines to baseline during illumination and render the fly blind. If you wait 1 minute, this is the light path. If you wait 1 minute in the dark, it will recover. So because of this transient nature of the receptor potential, I called it the transient receptor potential mutant, or in short TRP. And later on, this became the official name of the entire superfamily that I show you. Up to date, it was agreed by international committee. Okay, so it took me more than 15 years to figure out what is wrong with this mutant. When I came and became a faculty member in the Hebrew University, I did a very smart move. I collaborated with the late Tzvi Zellinger. And the reason that he wanted to collaborate is because from pharmacology, I found that a G protein is involved in the process that activates the system. And as you know, he was a world expert on G proteins. And people say that he should get the Nobel Prize together with the others who got it. But you know, there is no justice in these things. But he was a pioneer in G proteins. And he immediately, Gilman, you know, and other. And he immediately saw the power of using Drosophila. So this summarizes about 15 years of work. And we found by combination of biochemistry, I mean classical biochemistry, measuring enzymatic activity. I think soon there will be no biochemists because nobody measures enzymatic activity, as far as I know. It's all molecular biology. But he was a good biochemist. He was measuring enzymatic activity, light-dependent enzymatic activity, and I combined electrophysiology and pharmacology, and we figured out, against the dogma, by the way, because at that time in vertebrates they found that the system, the activation of the channels, the light-sensitive channel, is by cyclic nucleotide-gated channel. And everybody believes that evolution should work. And we found that this is not true for invertebrates. It works differently. It used a different cascade. And we found that this is a typical inositol-lipid signaling cascade. So evolution didn't work in vision. So it starts by rhodopsin that activate the heterotrimeric G protein, and this activates phospholipase C that hydrolyzes a minor phospholipid, PIP2, cuts it here into something hydrophilic, diacylglycerol, hydrophobic that remains in the membrane, and something hydrophilic, IP3. This one was known to activate PKC, but in our case it doesn't do it, it does something else. And the IP3 mobilizes calcium. And we had no idea what are the channels, and we had no idea what are the next steps. But we figured out that this takes place in the photoreceptor cell. These are light-activated reactions. And this was confirmed by others who isolated mutants. This one by the lab of Bill Pack, this by David Hyde, this again in the Bill Pack. And when you eliminate each one of these components, the fly is blind. So it was clear that all this cascade is essential for vision. But we had no idea what are the channels and what is the production of these second messenger, what is doing. And the turning point came by accident, as usual in science, when we could mimic the phenotype of wild type by adding lanthanum to the extracellular space. And we did it in 4 different species of fly. This is the first one. We tried it in musca, the house fly, because the photoreceptors were big. At that time, we didn't know how to do patch clamping on these. So we used intracellular recording, and you can see, again, historical slide. So this is the response to increasing light intensities in wild type, in the house flies that we kill all day, every time. And when we add lanthanum to the extracellular space, when we increase the light intensity, you see exactly the phenotype of the mutant. And it was reversible. If you wait some time, the lanthanum was washed and was partially reversible. If we do it to the mutant, this case, this is the homolog mutant of the TRP mutant in the bigger fly. This was also spontaneous mutation, was found by accident in Australia, by the way. And I got this mutant, we checked it. So lanthanum had no effect here. But it turned the wild type into mutant. So once I saw this result, I said, oh, now I know what TRP is doing. What is it? It must be a calcium channel. Because we know that lanthanum blocks calcium channel, and it fits with many small details that we found along the 15 years that didn't make any sense. But once you said, okay, it must be a calcium channel, then everything makes sense. And I was very excited. We published 2 reviews, and nobody paid any attention. Nobody was excited, and nobody cared about this. So I decided, okay, we have to give a more solid evidence. And at that time, Roger Hardy from Cambridge University, he introduced the patch clamping technique to Drosophila photoreceptors. So I invited him to come here, and we do. At that time, he was working on the synapse between the photoreceptors and the second order neuron. Said, well, why don't you work on phototransduction? He came here. We learned from him how to do patch clamping, and we did the compelling experiments comparing wild type to the mutant. And we show that what is missing in the mutant is the major light-activated channel that is highly calcium permeable. And the remaining response in the mutant, or after you put lanthanum, is the existence of another channel. And at that time, when we sent it to Nature, of course, it was rejected, lack of general interest, but luckily it was accepted in Neuron. And so now we have very important information. We know the cascade of activation. We know that this is a new type of calcium channel, and also the sequence was available, because independently, Craig Montel and Jay Rubin, at that time in Berkeley, they have a method to clone and sequence proteins. And they did it for many, like rhodopsin, and several of the rhodopsins, and also for the TRP. It was an exercise in molecular genetics. But the sequence was available. But it was clear that this is a membrane protein, but no one knows what was the function of this protein. And now we have also the sequence. So then we wrote another 2 reviews in teens, and people start to get interested in this, because these channels that we proposed was the target of the inositol-lipid signaling. And people were looking for years for the target of the inositol-lipid signaling. And there were many theories about this. And we offered them the whole solution. So people started to look on mammalian homologs of the TRP channels. And quite quickly, they found that this is a whole superfamily of channel proteins that covers, I would say, almost every aspect of life. So this is the TRPC's subfamily. The C stands for classical, because of the similarity of Drosophila. All of them were found because of the sequence homology to the Drosophila channels. And they, just to give you the feeling of them, they are extremely, there's a story behind each one of them. This one, the TRPC2, is essential for pheromone sensing. If you make knockout, mice cannot differentiate between male and female. And this mutation showed for the first time what pheromones are doing. For Yossi, the TRPC3 is very important in the cerebellum. Now there is a paper coming by Arthur Conner, and it's very important for motor learning. We still don't know why they need it, but definitely it's doing something very important. Because if you knock it down or you make it constitutively active, there is a very dramatic effect on the behavior of the mice. and so on. I mean, there are stories on each one of them. The TRPM subfamily, the M stands for melastatic, because the first one was found as a tumor suppressor of melanoma. And recently there was a very dramatic development in TRPM1, because it was found that this is the channel that is responsible for sign inversion in the on-bipolar cell. As you know, in our retina, they're all hyperpolarized, and there is a sign inversion in the on-bipolar. This is the channel. And now people try to find out how it works. And the TRPM5 is essential for taste. If you knock it out, the mice stop feeling bitter, sweet, and umami, the feeling of amino acids. TRPM8 is sensitive to menthol. It is activated by heat. It's also involved in pain mechanism, and so on. I mean, there is a story. I'll go back and talk about TRPM7 later on, but there is a story behind each one of them. They are all conserved from C. elegans to human, and also there are some versions in yeast. So it's a very ancient family. I'll concentrate on the TRPV because this will be kind of introduction to Alex. The TRPV subfamily, the V stands for vanilloid, because it was found by a screen by David Julius. This channel can be activated by the hot ingredient of chili pepper, capsaicin. So it was called the capsaicin receptor. And they are all also called the thermoTRPs, because they are activated by different gradient of hot temperature, and they control body temperature. Also, they are involved in burning pain and in many other mechanisms. I have no time to go into detail. Some of them are mechanosensitive. The TRPA1, the A code, I'll show you the structural feature because it has 18 anchoring repeats in the N terminal, and it is activated by spices, but also it is involved in pain mechanism and also mechanosensitive. These were found because of the hereditary disease. When this is mutated, it's called polycystic kidney disease. This is the mucolipidosis type 4, which is a lysosomal storage disorder. These are mechanosensitive. In short, I mean, I can give you 10 hours lecture about the striking finding about all these channels. And they are involved also in many diseases. They have overexpression in cancer. They are involved in kidney diseases, because these are critical proteins in control of many processes. And there is a lot of interest in them. Okay, but one thing, once you see that such a diverse family, you start to wonder, I mean, is there any common denominator among this family? And this question becomes critical, because when you can see how they can activate it, they can be activated, they are polymodal channels. They can activate by menthol, by capsaicin, by mechanical, by hyperosmotic solution. They can be enhanced by electrical activity, by cold, by hot, by mustard oil, by many spices, by receptor activation, by changes in the thickness of the membrane. And you wonder, I mean, why do you put them all in one superfamily? And so there is a common denominator to all these channels, and this is the structure of the channel. So this channel structure is typical for voltage-gated channels. It has a typical 6 transmembrane domains with a pore region between S5 and S6. The channel is a tetramer, like the potassium channel, and this is highly conserved, especially the pore region. But there are other parts that are different in different channels, and this gives a variability. I mentioned the anchoring repeat. In the trip A1, there is 18. In the trip, in the N, that I showed you later on, there are 27 anchoring repeats. Nobody knows why. They all have calmodulin binding site, and they have special features that are typical for different species. But the transmembrane domain is conserved. But unlike voltage-gated channels, the voltage sensor, the lysine and arginines in S4 are replaced by non-charged residues. So these channels are not voltage activated. And in fact, this is still a mystery how they are activated. And as I mentioned, we don't know how they are activated, not because that it is difficult to activate them. On the contrary, it's too easy to activate them. There is a lot of redundancy between the activations. And because they can activate it in so many ways, it is not clear what is the physiological mechanism of activation. And even the basic concept. Are they these ionotropic channels, like the nicotine acetylcholine receptors? Like David Julius, who found the V1, he called them the receptors because they were activated by capsaicin. So he said, okay, they are capsaicin receptor, they are ionotropic channels. But I told David Julius, look, this doesn't fit with what we know. We know that they are not receptors, they are effectors. They are the end of the cascade, activating by a cascade. Later on, he found that one physiological and maybe one of the most important way to activate them is via receptors, G-protein coupled receptors. And capsaicin is not a natural compound, but they can activate it by endogenous compound like anandamide, that is the target of that Rafi Mishulam found, the target of the endocannabinoids. Yeah. So it's not clear. I mean, what is the physiological way? And here, this is the importance of Drosophila. In Drosophila, we came from the fly. We know that these are the light-activated channels. These are the end of the cascade. But then we ask, can we activate them directly, like David Julius activated the trip V1 by capsaicin? And the answer is yes. And I'll show you. We can activate them also directly. But this is not physiological. But it has very important consequences on effects of disease. Now I'll go to show you, to compare between the Drosophila TRP channels and the trip V1. And the reason that I concentrate on the trip V1 is because of this recent development. There are two articles in Nature about the solution of the structure of trip V1 in the lab of David Julius. So this is a very useful tool. But when we started, and the student who did it is Maximilian Peters, who came from Germany, and he was trained as a pharmacologist, but now he can do everything. And by the way, he's now going to do his postdoc in the lab of David Julius, and he hoped to make also the structure of the Drosophila channels, hopefully, in the future, with the help from someone that...
Yifan Chen.
How is it?
Yifan Chen.
Yifan Chen.
Yeah.
Yifan Chen.
Co-corresponding author of the
Yeah, who was a structural biologist behind this important development of the structure. Okay, so the way that Max did was he used homology modeling. And at that time, he found that the best homology is the sodium channel. Although it has no voltage sensor, it's not a tetramer, but still the pore region of the sodium channel is most similar to the TRP channels. We thought at the beginning that this is more to the potassium and to the calcium, but not to the sodium. So he used homology modeling with the sodium channel, and he used bioinformatic techniques that I'll not go to this. And in order to validate his prediction, he used structure function. He expressed the channel with a point mutation and see if the prediction of the model fits with what he measured. Unfortunately, the major channel, the TRP channel, cannot be expressed in tissue culture cell. For unknown reason, we tried for many years, but so far we failed. There are a few papers claiming that they did, but they are all artifacts, including a paper in Cell. It's artifact, it's not true. And so what we do, we express the other channel, the minor channel that is less culture. It's non-selective cation channel, and this is called the TRP-like or TRPL. So all the experiment in tissue culture cells are in the other channel, because the major channel we cannot express. And so the basic technique is to take the TRPL. The TRPL is called TRP-like. It was cloned and sequenced by someone from Australia, and because of the similarity to the TRP, he called it TRP-like. And in the pore region, they are all virtually identical, except for one amino acid that determines the calcium permeability. And so we express them in hex cells, and in hex cells, you can see it here, this IV curve, it's not sensitive to voltage. The channel is closed, it's not sensitive to voltage. But if you activate it by PLC, so how we activate in PLC, we also express the muscarinic M1 receptors in the hex cells, and then we can activate it by carbachol. And then you can see that you get a nice current from these cells. And now we can, so we did a series of point mutation. I'll concentrate on mainly one mutation in the pore region that makes the channel constitutively active, and I'll show it soon. But this is I took it from the Nature paper of David Julius and colleagues. And mainly what is interesting is the pore region, the S5 and S6. And this is, you see, similar to other voltage-gated channels. And this is in the green is in the open state, and the red is the closed state. I use it because we make a mutation that makes the channel constitutively active, and we made other mutations, and we found that the prediction, just to cut a long story short, that the predictions from this model fits very beautiful to our prediction of the S5 and S6 of the TRPL. I mean, it's, the general structure is virtually identical, although the details of the amino acid is not identical. But the three-dimensional, from what you get, is, I'll show you the superposition. It's remarkably similar, which gives us hope that there is a common denominator between the TRP channels. The pore domain and the transmembrane domain is conserved. And even if there are differences in the amino acids, the basic orientation of the helices are similar, and the critical amino acids are conserved. And this is a good news, because we can use interchangeably and understand even diseases of point mutations, that we can draw conclusions from one subfamily to the other. So we concentrate, and I prefer to show you one study, because it was initiated at Purdue, at Purdue University, by a student of Bill Pak, Jae-Sung Yoon, who is now in Korea. We collaborated, but now he is more interested in industry and not, unfortunately, in science. But anyhow, there are, in the TRPL and TRP, there are 3 phenylalanines here, and you see they are conserved in the TRPC subfamily. They are conserved. Also, this is relatively conserved. And what Jae-Sung Yoon with Pak, and we collaborate on this, found that if you mutated this phenylalanine, actually it was a spontaneous mutation, then something very interesting happened. The channel became constitutively active. And this again was found randomly, but now we express it in the TRP, not in the TRPL, but now we express it also in the TRPL. So we express the TRPL in the hex cells, and with this mutation. So because we have to do a frame shift of 7 amino acids, but so the phenylalanine 550 in the TRP is 557. And the mutation that was found by Jae-Sung Yoon was the phenylalanine to isoleucine. Okay? And we found the same. So this is a wild type TRPL, you see no current. And now, without any stimulation, if we change the phenylalanine in the 5th transfer domain to isoleucine, then the channel becomes constitutively active. And so we try to see what happens if we change it to other amino acids. So in some TRP channels, the isoleucine is replaced by valine. So we look what happened if we use it with valine. We change the phenylalanine to valine, and the spontaneous activity is gone. So we can do it only with the isoleucine. But still, we could activate it by PLC. Although this constitutive activity is gone, if we activate it by carbachol, we express the M1, we could still get the current, and you can see it here. See PLC activation by carbachol, and we can block it from the lanthanide family with gadolinium. And we wonder, what is so special about isoleucine? Because it doesn't look like an important amino acid. And why only isoleucine is making the channel constitutively active? And now with our homology model, we came to the conclusion that the methyl group make a kink in the pore domain and make it a little bit straight, and this probably caused the constitutive activity. And the model of David Julius and colleagues confirms this, that indeed it's straightened, you change it. Yeah, so these are the different orientations that you have in the S5. And so when it's minor change, but we don't expect to see big changes. But it's fit beautifully and explained by the model of David Julius, that the open state become a little bit straightened. And this caused the constitutive activity, because David Julius and colleagues, they solved the structure in 2 states, in the closed state and in the open state by application of a toxin. And this is exactly the change that they see. So again, it fits beautifully. And the conclusions from all these structure function analyses is that the published structure of TRPV1 explains well our structure function study of the TRPL. And the TRPV1 and TRPL share identity general structure in the crucial pore region. And closed and open state of TRPV1 and TRPL share common structural features. And everything, all the mutations that we did, and we did many, all fits with all these conclusions. Okay, so now I want to move from this information to something more practical, to the experiments that we did together with Bill Pak and Jae-Sung Yoon. So the mutation that Jae-Sung Yoon found, there was a replacement of, in the TRP now, of phenylalanine 550 to isoleucine. And the reason they found this mutant that was sitting for more than 20 years in the collection of Bill Pak, nobody even dreamed that this is an allele of the TRP channel, because it didn't have any similarity in the phenotype to TRP. And it was degenerating, as you can see here. So this is the structure of a single ommatidium. And these are the peripheral photoreceptors. These are highly polarized cells. These are the microvilli where all the transduction machinery resides. This is the cell body. You can see it in lower magnification. Yeah, this is how you cut it here, and you do transmission in EM. This was done by Hagit Cohen in my lab. And this, what happens if you put one copy is not sufficient when it hatches, it still looks normal. But if you put 2 copies of the defective gene product, then you see very severe degeneration already in the pupa stage. And this was published in J Neuroscience several years ago. And so we wanted to mimic this situation of constitutive activity by other means, not by mutation, and also wanted to see if we can activate the channel without illumination at all, to mimic the situation of constitutive activity. And again, by accident, we found that there is a way. And the way is very simple, is to put the fly under anoxia. We simply blow, this you can do to fly. You can blow nitrogen on the fly, it becomes anoxic. Well, you cannot do it forever because it will die. But you can do it for 15, 20 minutes. And then there is something dramatic happens to the electroretinogram. So now we record from the electroretinogram. We put gross electrode on the eye, on the compound eye, and measure the ERG, electroretinogram. So here, this is a light stimulus. You give a light, and you get this ERG, corneal negative response. At the same time, I'll show you why, we also use a calcium selective electrode, and there is also a drop of this we found in different work, also drop in extracellular calcium, because these channels, the trip channels, are 40 times more permeable to calcium than to sodium. And when you illuminate, you open the channels, there is a large calcium influx that floods the photoreceptor with calcium. This is a later experiment. You can see it here. We use GCAM6. Okay, so this is indicators that you express in the photoreceptors. And when you illuminate, this is a mutant, but you see there is a very large increase in intracellular calcium, but at the same time there is a drop of extracellular calcium that you can measure with a calcium selective electrode. Now we put anoxia in the dark. So there is some artifact, and there is a complex waveform that we don't know what the origin, and only when you give the nitrogen, the anoxia. If you eliminate it, everything is reversible. But because there are 20 different cells in the retina, we wanted to record specifically, it's the time? Huh? So I have to, I'll speed up. So we wanted to see from what cells this comes from. And since the photoreceptors are the only cell that are calcium permeable, this is why we measure calcium. So we put the anoxia, and there is, you see there is a large calcium influx, reduction in extracellular space. So we're sure that this comes from activation of the photoreceptor cells of the TRP channels, because they are the only calcium permeable channels. And we wanted to know at what level, so we used mutants, blind mutants, for example, without phospholipase C, and we still could get it. So this is a late stage. And we could eliminate, we use the power of the molecular genetics of Drosophila, we can eliminate the TRP channel, we can eliminate both of them, and this signal disappeared. So it's clear that these TRP channels, and also TRPL, are very, very sensitive to anoxia. And so I'll just drop the electrophysiology. We showed that this is because of a reduction in ATP, because we could relieve this block, this constitutive, by uncaging of ATP. And the mechanism is not clear, but you see there are many phosphorylation sites on the C terminals. This is a work in collaboration with Armin Huber in Germany. And finally, what is the importance of this? I mean, and this I come to the more related to the topic of some of the talks. We know about glutamate toxicity in neurons. And this happens in ischemia, mainly stroke, trauma, or seizure. And there is accumulation of glutamate, and this initiates a positive feedback that causes increase in calcium, and this increase the damage and produce a reactive oxygen and nitrogen species. and cause eventually cell death. And because of the involvement of NMDA in this, people assume that if you use NMDA antagonist, you can relieve the symptoms of stroke. But this turned out to be complete failure, and it was not clear why. And so when we found that TRP channels can be activated by anoxia, by reduction in ATP, and TRP channels are heavily expressed in the brain, so this could be the initiation mechanism of this. And there was a study by Timiansky from Canada who found that if he, by RNAi, knock out TRPM7, then he could, which is expressed heavily in the cerebellum, in the hippocampus, this can strongly delay the onset of a neuronal cell death following ischemia. And now he's working for an antagonist, and we are also working for antagonists. I have no time to go, but we found that carvacol, the oil of oregano oil, is antagonist of the TRPM7, and so this is one way. So we think that following the pharmacology of TRP channels can help preventing neuronal cell death during ischemia. But this is a work for the future. And this is just one example how finding agonists and antagonists of TRP channels can be very useful for disease. And the take-home message is that TRP channels are polymodal channels that are activated by many stimuli and are involved in the function of almost every system of our body. TRP channels are involved in many diseases, and the gating mechanism of TRP channel is still an enigma. We still don't know how they are activated, but this is a good thing because it gives us work for the future. And thank you.
Quick questions? Just, would you say something about what's in the, going back to the very beginning,
Yeah.
when in the TRP mutant, what is the remaining signal?
The remaining signal is from the other channel.
From a different
Yeah, there are two, the TRP and TRP-like, and they are activated differently. So this is
I was wondering about the phenylalanine.
Yeah.
So do you see it in other channels now that you know that this is an important amino acid? Can you now go back and look?
if they are conserved also in voltage-gated channels.
Yes. For example,
Not in this, but yeah.
Switch.
Yeah, not in the same place. In the TRP channels, yes, it is. But in sodium or calcium or potassium channel, they are replaced. But the principle, I think, holds that if there is a kink there, this can affect the permeability of the channel. Yeah.
And I was also wondering why you couldn't express your channel. You said it led to cell death. I mean,
Yeah.
can't you just grow a cell line that expresses this to begin with? I mean, what is that?
I have to be more accurate. There is no problem to express it. There is a problem to express it functionally. It is heavily expressed. We see it with the antibody. But it doesn't reach the surface membrane for some reasons. That's a problem. And the transport mechanism of the TRP is something special because there are about 5 million channels in a single cell, and it shares the transport system with a rhodopsin. So there are 100 million molecules per cell. So this is a special transport system that needs to bring the, and we tried so many things, and others as well. It's not the only one. The trip of the pheromones, also people working on pheromones, Kathleen Durack, they are very much want to express it. And there are several stubborn TRP channels that you cannot express functionally. And I think it has to do, I didn't have time to go, these channels are also unique that when you activate them, they move. They can translocate from the plasma membrane to the cell body and back. And all the channels that are moving, not all of them, all those that are moving like TRPV1, no problem to express. But there are few who are bound to scaffold proteins like this trip. And all those who are bound to scaffold protein, you cannot express, which is a major difficulty. Yeah.
You emphasize that the trip channel are voltage insensitive. There are
Yes.
no voltage sensor.
Yes.
And yet all your voltage current curve are non-linear, completely non-linear.
Yes. Okay, this is a good question. This is a secondary effect of open channel block. All these channels, they suffer from open channel block. If you eliminate divalence, it becomes linear. But some of them, even if you do it under the divalent free, they show some voltage dependence, like TRPV1, and it's not clear where it comes from, because there are no charge residues in the membrane. So, but the Drosophila and many others, they are completely linear if you remove the divalent. It's an open channel block. And this is another story. What is the function of this?
No, but the non-linearity, the more they open, the more current you have. So how
Yeah.
open channel blocker fits into that? It should be the other way around.
Here, I'll show you. Oh. It's very strange. Okay, oh, it's stuck. No, it's. No, you're right. I mean, there's a point here. I don't want to take too much time. But in the physiological range, there is almost no current in the physiological range. And only when you come to positive voltage, you start to see the large current. And you're right. I mean, we wonder, I mean, how it can work. I mean, if in the physiological range, once the channel is open, immediately it's blocked by calcium or magnesium, like the NMDA.
But
And
it's block only in one direction of current.
yeah, in one direction. And we found the mechanism. This is a PhD of Shiko Parnas. That if you can do it, what light do is removal of open channel block for some time, because the PLC produces substances that remove open channel block without depolarization. And this is also true, by the way, for the NMDA. We found also that for the NMDA, you don't need voltage. With polyunsaturated fatty acids, you can remove the magnesium channel block. We found it, published in the same paper. Yeah.
Okay, good afternoon. Thank you very much for the invitation. I would like to start my talk with the definition of happiness. So happiness is having scratch for every itch. So now we know what's happiness. Today I'm going to tell you about what is itch, if it works. Yeah. So, and I would like to start by citing a Sidney Braddon column in a Current Biology, the loose end. In one of his first columns in around 80s, he was complaining about that all the neuroscience dealing with vision. And it's obvious why. It's easy to study. Everybody knows the tracks of the system. But then he suggested other areas for research and the other side. But it seems to me that the another area that has been neglected and which I think has great potential is the theory of itching, which is not a branch of Chinese metaphysics, but you plain common or garden itching that's common scratching, which also has nothing to do with Taoism. I know that I scratch an itchy patch because it gives me relief from the demandingly irritating sensation. I know I do it consciously because an itch, like pain, grabs you, and viciously itchy mosquito bites can even wake you from a deep sleep. I'm also certain that all this subjective knowledge is shared by thousands of co-subbers. So we're in the lab studying pain. For me, it was needs some more evidence to convince me that itch is also important. So I went back to the history, and in the book of Job, one of the punishments of Job was the horrible scratching, and then Job took a piece of broken pottery and scrubbed himself with a set among the ashes. And even in Inferno, okay, one of the very advanced circles of Inferno, people there were punished by itching. So it's pretty horrible sensation. And then, if you're going back to Sidney Brennan, basically on the continuation of this column, he basically writing a grand proposal about how we should study itch. And to summarize it, this question that he and then we asked it, are there specific itch receptors in the skin? Are there specific itch fibers in the skin? Are there labeled lines for different types of puritogens that are agents that produce itch? If all the above are correct, can we selectively block itch? And so what I'm going to talk today, it's one of the projects in my lab that I did together in collaboration with Clifford Wood from Harvard Medical School. And this work has been done by David Robertson, my former PhD student, and Sagi Gudes. And this story basically defines that we do have a labeled line for different type of puritogens. So each type of puritogen or each type of itch, it's transferred or transmitted by very selective neurons, and we can selectively block this type of neurons. Okay, so what is itch? And as Sidney Brennan mentioned Chinese metaphysics, and correct me if I'm wrong, but according to Chinese metaphysics, itching represents dissatisfaction with the way things are going, or dissatisfaction with yourself. So if your paper is rejected, just start scratching. Okay, but from the neurobiological point of view, itch is a complex, unpleasant cutaneous sensation that in some respects resembles pain, yet in difference in terms of intrinsic sensory quality and urge to scratching. Okay? So the first question I would like to ask, are there specific itch receptors in the skin? But maybe before that, I should ask, what are the agents that produce itch? What are those puritogens? And there are plenty. The most known or most famous is histamine. But apparently there are plenty of non-histaminergic factors that will also produce itch. And we'll talk about chloroquine as one of them, but there are some others which is very, very interesting. And so coming back to the question, if we are looking on some scheme that we created of peripheral terminals in the skin and with all the mast cells around, we know that there are several receptors, specific receptors for puritogen. For example, histamine will activate H1 receptor, which sits on peripheral terminals. Chloroquine, which is another non-histaminergic type of puritogen, will activate another G-protein couple receptors called mass-related G-protein couple receptors A3. But those are G-protein couple receptors. So activation of the receptors cannot produce an activation of neurons. So they must have some inotrophic receptors. And interestingly enough, that connects us to the Baruch talk. So histamine via PLC cascade will activate TRPV1 channel. And just to remind you, TRPV1 channel, it's used to be a keystone for pain neurons. It's activated by noxious heat, by low pH, and by capsaicin. But it could also be activated indirectly via PLC cascade, and histamine could be part of this cascade. So activation of TRP channel will produce a prominent inward current, depolarization, action potential, and signal to scratch. So chloroquine will by activation of mass-related G-protein couple receptor A3, will activate another channel from the TRP family, TRPA1. And again, just to remind you, this channel also is the landmark for pain neurons. And normally it's activated by mustard oil, wasabi, cinnamon oil, noxious coat, and maybe some other, and a lot of noxious chemicals like paraformaldehyde. Interestingly, this TRPA1 channel, it seems to be a key molecule for all non-histaminergic or puritogens. And it was a very recent paper in Cell, published a couple of months ago, showing that explaining its key role in the mechanism of allergic itch in atopic dermatitis or eczema. And just to make a very, very long story short, all these allergens activate keratinocytes that then release a factor called TSLP, which activates this receptor, and this receptor will activate again TRPA1 channel to produce electrical signal and scratching. Okay, so what we know till now that there are several different receptors, so that we have specific puritogens, and all of them activate specific receptors. But the main question remains, and then one of the main questions actually, is whether we have one master each neuron that will express all these receptors, and all these puritogens will activate one single type of neurons, or we do have a labeled line. We do have a specific neurons for histamine each or a non-histaminergic each. Okay? And so basically this is summarized the question. Do we have two separate neurons, one which will activate by histamine, will also express TRPV1 channel, and second one will be activated by chloroquine or TSLP, and we will express MRGA3 receptors in TRPA1 channel. And there are several options in the literature, but I must say the jury was still out before we came with our approach. So this is the one option, and this option basically shows that in young neurons, there are some neurons that will respond, and this is calcium imaging time, and this increase in fluorescence will respond to application of chloroquine. And again, we can see calcium imaging as a response to chloroquine because activate TRPA1 channel, which is calcium permeable. It will also respond, same neurons will respond to histamine, calcium flux via TRPV1 channels, and to capsaicin, meaning that we do have a one master neuron that expresses both receptors for histamine and for chloroquine. It could be activated by both. A later heroic study by Dong group showing in vivo recording from DRG neurons that they demonstrate that the same neuron will respond, and this is a action potential, by histamine and by chloroquine and by capsaicin. Again, one master each neuron. But there are some other evidence showing that in more elder animals, the clear difference between neurons that respond to chloroquine and to histamine. And again, in the same model, in vivo model, there's a neuron that will respond to beta-alanine, another non-histaminergic puritogen, but not to histamine. So we still don't know what happens. Another very interesting question is, since TRP channels, TRPV1 and TRPA1 channels, as I mentioned, are key molecules that basically that I identified pain neurons, could it be that pain in each are the same peripheral neurons, and the decision about pain in each happens later on in the level of spinal cord? Okay, so all these questions, basically, there was no answer. But then we have developed a new technique, a new approach, in which we could selectively silence active neurons, and then we can actually explore the function or loss of function of specific neurons and understand their role in pain and each sensation. I call this technique charge and target, because we use charged form of sodium channel blockers, and we target this charged form of sodium channel blockers selectively into either pain or itch neurons. So how it works? Just to remind you that local anesthetics like lidocaine, when injected into tissue due to physiological pH and their specific pKa, basically create two forms, a charged form, constantly charged form, and uncharged form. And only uncharged form is active because the binding site for lidocaine it's on cytosolic part of sodium channel. So it penetrates the membrane, that create again a balance between charged and uncharged form, and both of these form block sodium channels. So this charged form is an effective, and uncharged form is basically effective, and it's worked perfectly. The problem is that uncharged form could penetrate via any membrane. So if it's penetrate via membrane of the pain fibers, it will block pain, which is good, very good. But if it penetrate the membrane of alpha motor neurons, it will produce paralysis. Or if it's sensory, it will produce sensory block, and so on. So our purpose initially, when we started to work on selective pain blockade, were to create an approach that will allow us to block selectively pain neurons without affecting any other neurons. And our idea was, what if we will use constantly charged form of lidocaine? Okay? So this molecule, known by name QX314, it's permanently charged form of lidocaine. When we apply it from outside, it's ineffective. It cannot block sodium channels because it can penetrate the membrane. So maybe we will have a way to selectively target this molecule into pain neurons. And our idea was, since the TRP channels are expressed selectively on the pain neurons, maybe we can activate them with capsaicin, for example, chili pepper, or with mustard oil, and allow the penetration of this charged molecule, charged lidocaine, via the pore of the channel to the pain neurons, and then it will produce pain selective blockade. We thought about it because these trip channels are a cation non-permeable channels, and with very high permeability. We now know that they allow penetration of molecules up to 900 Daltons. Okay, so a charged lidocaine QX74, it's around 450 Daltons. So we were pretty theoretically it might work, and we actually check it. So the first question we ask whether application of this charged lidocaine and capsaicin to open the channel will allow the penetration and blockade of sodium current. And as you can see here, this is recording from cultured DRG neurons. The small cell normally are pain-related cells, and large cells are also sensory cells, but are not transmitting noxious information. So when we record from small neurons, and this is time, and all those are compressed a recording of sodium inward sodium current. When we apply capsaicin, it creates depolarization, and therefore there are some transient decrease in sodium current amplitude. But if you compare a sodium currents before application of capsaicin and after application of capsaicin, there is no difference whatsoever. When we apply QX74 from outside, also there was no difference whatsoever, again because QX74 cannot penetrate the membrane, so it's not active. When we apply capsaicin with QX, sodium current was almost totally blocked. Okay, and interestingly enough, when we compare the effect of capsaicin QX on large neurons, which are not expressing TRP1 channels, there was no effect of capsaicin QX on sodium currents, which is so basically we were able to selectively block sodium currents in small or pain-related neurons. We then continued and checked what happens whether this blockade will be sufficient to block action potential generations. And as you can see here, application of capsaicin QX totally blocked a generation of action potential in capsaicin sensitive or pain neurons, and has no effect whatsoever on action potential generation in sensory neurons, which is not transmitting pain information. So it very nice in a single cell level. We then move to the behavior study, and this is not the original paper, the paper that follow up our paper. So this is the typical graph that shows the changes in the behavior with time. It's called pore withdrawal latency. In this case, we apply a laser beam to hit the pore up to 52 degrees, and then after around 14 seconds, animal will remove the pore. So basically, we check the withdrawal reflex of painful stimuli. And as you can see here, application of QX314 have no change whatsoever on the reflex. Application of capsaicin produced some decrease, hypersensitivity, as expected. It's very painful. Application of capsaicin QX remove the ability of the animal or make the animal totally insensitive to this painful stimuli. Okay, the same happens with mechanical painful stimuli, but interestingly enough, neither light touch, not motor activity were affected. So on the level of the whole animal, we were able to create or to produce pain selective anesthesia. We then move forward and we're now performing experiment in large animals, and this is recording from pigs. This is a local nerve block. We also did some epidural anesthesia, and this is a comparison between lidocaine and capsaicin QX. Black is pain, gray is motor activity. When we apply lidocaine, as we expect, we block both. 2, it's normal. 0, it's totally blocked. We block both. When we apply capsaicin QX, we block pain, but not motor activity. Okay, so when we activate TRPV1 channel, this activation is sufficient to produce the entry of QX314 and selectively block sodium channels. Thus, we will be able to create pain selective anesthesia. As I mentioned, but for each, we need both. We need TRPV1 for histamine and TRPA1 for non-histaminergic receptors. So our next step was to prove whether activation of TRPA1 receptors will also be sufficient to allow the entry of QX314. Okay, we can activate TRPA1 channels with something called AITC or mustard oil. So we apply mustard oil, and it activates the channel. And the same experiment that we did with capsaicin, in this case mustard oil, sodium currents recording, application of a mustard oil with QX314 eliminate sodium current and also eliminate the hyperalgetic effect of mustard oil when injected. So we have a solid basis to move forward and now ask questions about each. So what happens in each is a little bit different. With the pain, we were able to block pain by activating these TRP channels. But during each, these channels are already active. Okay? the puritagens will activate these channels, and therefore we feel itch and we scratch. So maybe we don't even need to apply capsaicin or mustard oil to open the channels because they're already open. So the question that we ask whether endogenous activation of TRP channels will be sufficient to allow entry of QX74 and block the activity of the neurons and therefore to block itch. So we perform some experiments first with cultured trigeminal ganglion neurons. And the reason we move from DRG, from dorsal ganglion to trigeminal ganglion neurons, it's because later on we inflicted itching or scratching by injecting the puritagens in the area of the face. So we just check the corresponding areas. So you can hardly see it, but there are some neurons, and we apply histamine first, and then some of the neurons, about 4% of the neurons, respond to histamine. Again, we can see it by elevation of calcium, and the blue one was non-responsive, non-sensitive to histamine. We then, from these neurons, recorded sodium currents in control condition and after application of histamine and QX. And again, the idea, histamine will open TRP channels, QX will penetrate in and will block sodium currents. And it's exactly what happens when we record from the red neuron, which is histamine sensitive or histamine positive neuron. Application of histamine QX totally blocks sodium current, but not when we record this, we apply the same histamine QX on this neuron, which was histamine negative. And all this blockade of selective blockade, I must say, of sodium currents in histamine positive neurons was prevented by application of specific antagonists of TRPV1 channel. So puritagen mediated activation of TRPV1 channel is sufficient to block a sodium current. And the same happens when we apply chloroquine. Again, chloroquine will activate TRPA1 channel, and idea was that QX will penetrate and block sodium channels. And again, what happens? This neuron was about, again, 4 to 5% other neurons that we record from were sensitive to chloroquine. We record from this neuron, and application of chloroquine QX block sodium currents, but not from neurons that were not sensitive to chloroquine. And again, this effect was abolished by application of specific TRPA1 antagonist. So theoretically, it might work. So our next step was, okay, let's do a clinical work. Let's check if we apply QX via a histaminergic scratching or histamine scratching, would it block itch or not? And so we apply histamine, and then we apply histamine with QX. And as you can see here, so this is time, and this is number of scratching bouts per 1 minute. Okay. So for our great disappointment, application of histamine QX did not change a lot scratching behavior. So we were disappointed, but then we realized that QX74 is a charged molecule. So it takes some time to penetrate and to reach the nerve and then to act. So we changed our model. So what we have done, next again, we inject the puritagen together with QX74 first. We inject it intradermally to the cheek of the animal, because it was published that when we inject to the cheek, it was easily distinguished between itch, which produces scratching, and pain, which produces wiping. Okay, so we injected intradermally puritagen, let's say histamine with the QX, waited for half an hour, and then injected the same puritagen again, and we measured scratching. Okay? Yeah. Okay. So just to be sure that a second injection of puritagen will not produce any desensitization, we inject histamine and histamine again, and animal scratches perfectly. We then inject histamine QX, waited, and when we inject histamine second time, scratching disappeared. Meaning that the application of histamine QX allowed QX slowly penetrate to the nerves. It blocked sodium channels. So second injection of histamine, although activate histamine receptors and TRP channels, will not produce any behavioral side. Okay, same happens with chloroquine. So when we apply chloroquine and QX and waited, and then chloroquine again, the number of scratches was reduced dramatically. We then just for fun used another non-histaminergic agent called SLIGO, which is the peptide, itch-induced peptide, and when we applied SLIGO, SLIGRL peptide with QX, and then this peptide again, was no scratching anymore. So again, theoretically, we do have a very good approach that potentially could help these people that suffer from chronic or even acute itch. Okay, but then we would like to ask, and this is just summarize the findings showing that activation of each receptor will produce enough activation of trip channels to allow QX to penetrate and block a sensation. This is from Newsom views from Diana Bautista. Okay, so this was again clinical, quite clinically relevant, but then we can ask some more scientific question. And again, the first question that I would like to ask, whether itch and pain are similar or not. Would I have a specific system for itch and specific system for pain? And the way we address this question is just by blocking all itch-related neurons, by either applying histamine with QX or chloroquine in QX, and then examining behaviorally whether there are some changes in the pain sensation. And so what we have done here, we injected capsaicin or mustard oil into the cheek and measure wiping. Again, it's painful, so animal will wipe the cheek. And this wiping, capsaicin-induced wiping, was totally blocked when you inject capsaicin QX. Same reason as explained before. Capsaicin will open trip channels, QX will penetrate and block pain, but was totally the same as a control when either histamine QX or chloroquine QX was pre-injected. Same happens with mustard oil wiping, meaning basically that we don't need itch neurons to feel pain, and we did again this experiment, now injecting histamine QX or chloroquine QX into the pore and measuring responses to painful stimuli, and again mechanical pain, thermal pain, cold pain was unchanged, or response to this stimuli was unchanged following blockade of itch sensing neurons. And also I must say we also checked the normal sensation, okay, and also was unchanged. So what we can conclude for this, we cannot say for 100% that the pain and itch are separate, but we can say we're pretty sure that a functional proreceptors, or these itch sensing fibers, are dispensable for normal pain sensation. Our next question was to ask whether we have labeled line for histaminergic itch and chloroquinergic itch, or we have this master itch neuron. And the idea was as follows: let's inject histamine QX, and as I showed you, histamine QX will block all histaminergic fibers, because a subsequent injection of histamine will not produce a scratching. If you then, instead of injection of histamine, we inject chloroquine, if animal will stop scratching, meaning by blocking of histaminergic itch fibers, we'll block whole itch fibers. But if animal will scratch again, meaning that histaminergic fibers and non-histaminergic fibers are separate. And that's exactly what we did. Again, we injected puritogen X with QX314, and then we inject puritogen 1. Again, a little bit different way of putting the data. So we here examining the response for histamine scratching following injection of vehicle, and as you can see, we inject the vehicle, and then histamine, animal will scratch about 50 times in 5 minutes. We then injected what happens with this histamine scratching when it was injected after either histamine with QX or chloroquine in QX. And as you'll see here, injection of chloroquine in QX will not affect histamine scratching, and vice versa. Injection of histamine QX will not affect chloroquine scratching. Basically, if I inject histamine in QX and then chloroquine, animal will scratch as well as scratch it when we inject vehicle. Basically, it means, interestingly enough, when we inject a histamine QX, it will not affect a Sligo scratching, another non-histaminergic type of scratching, but when we inject chloroquine QX, it will block totally scratching as a result of Sligo. Okay? So what we can conclude from this data is that we have separate functional fibers for a sensing or a detecting histaminergic itch, and separate fibers for detecting non-histaminergic itch, which in this case present by chloroquine and Sligo, but we now know that it's much more, so these fibers will transmit information about much more non-histaminergic fibers. Okay, when we then, just to be sure, we went back again to the level of the cells in the culture, just to prove it more molecular level, and we saw that about 4% of cells respond to histamine, about 3% of cells respond to chloroquine, with almost no overlap. And another interesting piece of data showing that most of the cells that respond to histamine will also respond to capsaicin, and most of the cells that respond to chloroquine will also respond to mustard oil. meaning that, as we already know, histaminergic fibers will express TRPV1, and non-histaminergic fibers will express also TRPA1. Just to be sure that it's indeed so in the model that we examined, we examine it by injecting algogen, either capsaicin or mastodol with QX. Again, it will block all either TRPV1 or TRPA1 containing fibers, and then we inject puritagen. So, and again, we are measuring scratching. So we inject either capsaicin QX or mastodol QX and measure how much animal will scratch as a response to histamine. So again, vehicle and then histamine, about 50 scratches in 5 minutes. Capsaicin QX, which block all TRPV1 containing fibers, totally block histamine scratching. Mastodol with QX will block all TRPA1 containing fibers, have no effect on histamine scratching. The picture was a little bit different with non-histaminergic itch. And again, now measuring chloroquine scratching, chloroquine-induced scratching, or Sligo-induced scratching following an injection of capsaicin QX or ATC QX. So basically, ATC QX totally blocked this chloroquine scratching or Sligo scratching, while capsaicin QX reduces significantly. So what we can conclude that the histaminergic itch is totally dependent on the fibers that express TRPV1, whereas a chloroquinergic detecting fibers express TRPA1, but some of them also express TRPV1. Okay, but then just by chance we found another very interesting data. So what we have done here, we injected algogen, capsaicin with QX or mastodol with QX, and then capsaicin or mastodol again. But in this case, we instead of examining pain, which I explained to you before, we check what happens with the scratching. So normally, when we inject capsaicin or mastodol, it will produce only wiping and no scratching whatsoever. But when we inject mastodol with QX and block all TRPA1 containing fibers, and then inject capsaicin, animal was both scratching and wiping. So by blocking the TRPA1 containing fibers, we unmasked scratching sensation as a result of injection of capsaicin, and vice versa. When we inject a mastodol, animal will normally only wipe because it's painful, but when we block all TRPV1 containing fibers by injection capsaicin QX, animal will also scratch. So basically what we are claiming from this data, that there are specific population of peripheral neurons that contain both TRPV1 and TRPA1, and activation of these neurons will produce central inhibition of a sensation of itch. And this is basically the one of the summary slides, that this is the population of peripheral neurons. So we have the TRPV1 and TRPA1 containing neurons. Neurons that contain TRPV1, some of them will also express H1 receptors, and neurons that contain TRPA1, V1, some of them will also contain receptors for chloroquine. And there, again, some population of neurons which will express, activation of TRPV1 receptors, this red population, okay, will produce pain, okay? Activation of this population, so I'm colorblind and trying to find myself among all these colors. So activation of these fibers, okay, which is a TRPA1 and a receptor chloroquine, will produce itch. So I can say for sure that we have different central neurons for pain for itch from some previous data that shows that we can eliminate some type of cells, in this case gastrin-release peptide receptor cells, by using diphtheria toxin, and we will eliminate itch sensation but not pain sensation. So on central level there are two separate pathways for pain and for itch. So again, activation of red one will produce pain, activation of this one, the yellow one, I assume, will produce also pain. Activation of mixed one will produce itch, but activation of this neuron that contain both TRPV1 and TRPA1 will activate a inhibitory interneuron, the level of dorsal horn, that will inhibit sensation of itch. Okay, and just to sum up, just to sum up, this is the cover image we suggested to Nature Neuroscience, but they decided not to take. I don't know why, but I'm pretty proud of it, so I'll show it to you. So our findings support the presence of functionally distinct set of peripheral fibers, each of them responsible to transmit information about particular type of perithogen. Okay, by doing a targeted silencing of this neuron, we can differentiate between pain neurons and itch neurons, and we also find out specific peripheral population which basically acts to mask sensation of itch when we have painful stimuli. And we believe that our approach it's very clinically relevant to treat both acute and chronic itch. And just to thank you to my collaborators, this is a student that make it work in my lab, to Clifford Spoolflap and Professor Och from Seoul National University, who together with me sit for days and nights to count these scratching bouts. And this is members of my lab, and this is the founding agencies. Thank you very much. Questions? Yeah.
So, very nice work.
Thank you.
I was wondering, I mean, you elegantly showed that when you activate the TRP channel, you can induce the entry of a charged Q,
Yeah.
X, whatever compound.
Charged lidocaine. Yeah.
But I was wondering about the other consequences of opening this channel, because there are other compounds that may enter together, especially charged compounds that you might not want to, you know. So, I mean,
Okay.
you're only measuring scratching, but what are the other potential consequences of just opening these channels?
Okay, so we're measuring scratching and pain. So let's start with, let's be positive, for example. So the opening of these channels are pretty much expressed only by peripheral neurons. Okay, so when we inject these compounds peripherally by creating a nerve block, we're pretty sure that it will affect only pain fibers or itch fibers. What we know now, as Baruch mentioned, there's some TRP channels, similar TRP channels on tumor cells, on cancer cells. What we're now doing, we're activating these channels on tumor cells, and then applying charged cytotoxic compound that penetrates the tumor cells and selectively kill them. Okay, so this is the positive consequences. About the negative consequences, so we as both for pain and for itch, we look for the animals for a very, very long time and nothing happens. But you're right. It could enter. Stuff could exit from the cells. So we believe actually that QX314 that enters the cell. Actually, we show it that QX314 that enters the cell via these channels will also exit the cells via these channels. So it could be anything. But yeah, yep.
So conceptually, why do you think there are two parallel pathways for itch, for histaminergic versus non-histaminergic?
This is a very good question. So first of all, maybe we should think what's the evolutionary value of scratching. Okay, and evolutionary value of scratching, and not for itching, because scratching is a response, it's to remove the parasite. So we have a sub-parasite that will create itching, and we'll mechanically remove it. And actually, this mechanical pain of scratching will block itch in the central level. But whether it's now important to differentiate between histaminergic itch, non-histaminergic itch, I don't know. I think that people that feel itch as a result of mosquito bite or eczema have pretty much same sensation. So maybe we just left it with from all the time. Maybe we can discuss something in non-dentals and find out what's the reason.
So also along the same, similar thing, you have the same neuron that can encode both pain and itch, right?
Yes.
So how? If the neuron integrates at the level of the cell body and then just transfers the information upstream, how? If it fires, it fires. How can it differentiate?
No, we believe that we have a separate population of neurons that encodes pain, which is either that express either TRPV1 or TRPA1 or both. There are specific neurons, which is pretty limited population, 4% as I show you, that encode itch. These neurons could be theoretically activated also by capsaicin or by heat or mastodon. But for this activation, we do have this additional population of neurons that will also be activated by painful stimuli, but via interneuron, and we know what this interneuron will block itch and will mask itch and will not touch pain. So normally, in general, when we have painful stimuli, we have massive activation of pain neurons and some small activation of itch-generating neurons, but each is masked by activation of this additional population that inhibits it. Yes.
So are the itch and pain coded by separate multi-acellular or singular. I just wonder whether this evidence showing that each pain are codes for different neurons in the brain.
So actually, this is a very good question, and we looked or tried to look in literature that deals with higher processing of pain in each, and we cannot find a lot. So there was paper. First of all, we know for sure that at the level of dorsal horn, there are two separate populations. One encodes pain or transmits pain, and second one transmits each. What happens then? We don't know. I think that the level of thalamus, it was shown it was Nature Neuroscience paper about 6 years ago that shows that there's a separate population of neurons and one can inhibit other. On more higher level, I just don't know. I don't think there's the literature on this topic. And actually, I must say that even for pain, we're not 100% sure what encodes pain in the brain, or at least in the cortex. Yep. Okay, thank you very much.